Hello, everyone. Thank you, Freya, for the awesome introduction. So again, my name is Stephanie Kawecki. I'm a PhD candidate and New Harvest Research Fellow currently working in the Rowett Lab at UCLA. And so if you couldn't tell already, I'm definitely from Los Angeles. I think I'm the only person in the room wearing sandals today in the middle of fall. But today, I'm excited to talk about fat and how we can use tunable and edible scaffolds to engineer adipose tissue and potentially increase the efficiency of cultured meat production. So I think I can also skip this slide. When I first started working in cultured meat, a lot of the previous studies focused a lot on muscle tissue growth. And so I felt like I had to convince people about why we should focus on fat. So it's great to see all of the research that's occurring with adipose tissue engineering. But essentially here, I just wanted to highlight how we know that intramuscular fat is important, and we use the levels of intramuscular fat to grade our meat all around the world, because essentially people are willing to pay for meat that has higher levels, given that it improves sensory profiles such as flavor, texture, and tenderness. So I'm just giving some shout outs to some other adipose tissue engineers that are here with us. So we're already seeing some great approaches in the field of cellular agriculture towards engineering adipose tissue. So in this example, Dr. Yuen and others in the Kaplan lab developed an approach to culture adipocytes on 2D substrates and then aggregate them post-differentiation using alginate as a binder. And as you can see in this approach, that they're able to beautifully differentiate these porcine adipocytes in reduced serum environment on the timescale of 30 days. I won't go over this example. We just heard beautifully from Johanna about this. But in this approach, I did want to highlight how they were able to engineer adipose tissue further in a serum-free environment, and that they also use alginate as a hydrogel scaffold to culture their cells, again, in the timescale of around 28 days. So while we're seeing different approaches, both without using scaffolds and using scaffolds, our lab is really excited about the potential to use scaffolds to support the scalability of cultured meat. And some reasons why is we feel that it can contribute to the product volume, the texture, and potentially the nutrition. For example, we can create novel products like meat that's high in fiber. Doesn't that sound delicious? We can also introduce porosity using scaffolds, which can increase the diffusion of oxygen and important nutrients into the innermost sections of our tissues, allowing us to engineer thicker pieces of meat. And scaffolds can provide structure for organized tissue. This is especially important when we're trying to make complex tissue like a marbled cultured steak. So in my previous work, I developed scaffolds and an approach to engineer marble cultured meat. So in this approach, I engineered electrospun aligned nanofiber scaffolds, given that the alignment of substrates have shown to drive the differentiation of myocytes into myotubes. In parallel, I then fabricated microbead scaffolds that were more compliant, given that softer substrates have been shown to promote lipid accumulation in adipocytes. Using this approach, I can then seed my cells on their respective scaffolds and then differentiate them in their preferred medium and stack them post-differentiation to form multi-component tissue or marbled cultured meat. And what was most interesting with my study is that I found that the layers are actually adhering to one another on the timescale of hours without the use of additional cross-linkers. And upon subsequent mechanical analysis, I was able to confirm that it was forming a mechanically stable tissue product. Oops. So here's a cross-section of my multi-component cultured meat. On the left is my tissue versus a Wagyu beef steak, so I definitely set the bar high for myself in terms of comparison. But what I found is that while there's room to optimize the thickness of muscle tissue layers in my cultured meat samples, I was able to successfully replicate the length scales of adipose tissue compared to conventionally produced steaks. Taking a closer look, here is an image of primary rabbit skeletal muscle cells that were differentiated in red on aligned nanofiber scaffolds, and then layered with primary rabbit adipocytes that were differentiated on these cyan beads. And so what I found is not only are these layers self-adhering to one another, there also appears to be an integration of the two layers. So overall, while I was working on this project, I found that the utilization of scaffolds allowed me to really tailor the environment for each cell type and then subsequently engineer multi-component tissue that's organized. And lastly, we're really interested in scaffolds given that it can provide mechanical stimuli to cells. So within the field of mechanobiology, there's a whole body of literature that goes into mechanisms and describes how cells respond to their mechanical environment. And so, for example, we can arrange an environment so that the cells have a smaller adhesive area. Sorry, I'm working this pointer. And a softer substrate. And in these examples, it causes the cells to have a more rounded morphology and less cell spreading, versus providing a larger adhesive area or a stiffer substrate where you can actually increase the spreading in cells, resulting in higher cytoskeletal tension. And studies show that these mechanical environments can actually stimulate certain biosynthesis pathways similar to soluble growth factors. In this study, they seeded 323L1 mouse adipocytes within alginate hydrogels of different stiffness. And what they found is that there was higher levels of lipid accumulation in the adipocytes when they were grown in a more compliant substrate around 3 kilopascals, for example. Another way that we can affect the mechanobiology of cells is through cell density. So whether you have high cell density or low cell density can affect how they behave. And a potential mechanism as to why this is occurring is through this YAP-TAZ complex. And what's unique about this complex is that its location within the cell can alter depending on the mechanical stimuli on the cell. So for example, in these environments where you have more stiffness, more spreading, it can actually stretch the cells, stretch the nucleus, and allow YAP-TAZ to infiltrate into the nucleus, and then activate certain transcription factors that can affect proliferation, differentiation, and even apoptosis. Specifically with adipogenesis, studies have found that the inhibition of YAP-TAZ or its localization within the cytoplasm can actually promote adipogenesis. So we've seen studies with human stem cells that its inhibition of YAP-TAZ has shown differentiation into adipocytes. And then in ovine adipocytes, we've seen an increase in lipid accumulation with the inhibition of YAP-TAZ. So it was studies like these that really guided my first work in terms of how I could engineer my scaffolds to cater towards adipocytes. So with these microcarriers that I developed, I tuned them to be a stiffness of around 3 kilopascal. I also referenced biomedical literature to tune the size of my aggregates to be around 60 to 100 micron in diameter. And in fact, this is the size range, even a little bit larger, around 200 micron in diameter, that a lot of commercially available beads are at. So next, I seeded primary rabbit cells and mouse cell lines onto these scaffolds, and I tracked their lipid accumulation over time. And what I found is that lipids were accumulating on the time scale of weeks. And we see this with a lot of other adipocyte studies as well. And so then I started thinking, if we really want to increase the efficiency of cultured meat, it could be beneficial to increase this or speed up this process to have lipids accumulate faster. So then I started thinking about potential ways to accelerate adipogenesis. So we went over one way, talking about softer substrates, but then there's also higher cell density and smaller adhesive area. So then this gave me the idea of tuning microcarrier size to facilitate these interactions in adipocytes. And then I looked into the literature and found that there actually isn't not a lot of whole literature that describes the effects of bead size on adipogenesis. So to begin investigating this, I used our previously described protocol to fabricate gelatin microbeads using water and oil emulsions and cross-linked with transglutaminase. I can then size filter them to have beads of different size ranges, ranging all the way from less than 20 micron in diameter all the way to 150 to 300 micron in diameter. And here is the quantification of my beads, just to ensure that I was getting beads within the appropriate size range. But another important thing to note is that you can tune the amount of bead ranges that you're getting. So in this example, I'm spinning my emulsion at 550 RPM, and I'm seeing higher amounts of beads within the less than 20 micron diameter range. So this can be tuned. So next, I took my beads of different size ranges and I seeded primary rabbit adipocytes on each scaffold type. I also had samples that had no beads, so just forming aggregate spheroids. And what I found when I quantified the lipid fluorescence intensity per cell in all of my samples is that there was a significant increase in lipid fluorescence intensity when I seeded cells on microbeads with diameter less than 20 micron. And so with this data, we're really excited to explore molecular mechanisms next, as well as transcript levels of adipogenesis markers. It is interesting to see that with the high cell-cell contact that we're seeing without scaffolds, as well as the lower diameter scaffolds, we're still seeing an increase in lipid fluorescence over time. And so it makes me think that along with cell-cell contact, perhaps there is some benefit to having cell matrix interactions as well to induce adipogenesis. We're also translating this approach using porcine adipocytes. So thank you very much to the Kaplan Lab for generously providing these cells to begin working with. These studies are in the preliminary stages, and so we can't do any comparisons just yet. But I did like to highlight that we were able to form spheroids with these cells, which was very exciting. And then also just noting the amount of lipid accumulation that we're seeing in these porcine adipocytes within 3 days. So I'm sorry to leave you all with a cliffhanger there, but hopefully this will be published soon. We're also working towards animal-free microbead scaffolds, and so I pretty much translated our gelatin approach using pectin. And we are acquiring pectin from CP Kelco, if you've heard of them. They're a large food company, and they have a lot of their products in plant-based alternatives. And so here what I'm doing is I'm adding the pectin. I'm doing a similar emulsion, but instead adding calcium carbonate to cross-link. And in this process, I'm able to form microbeads. And then I developed an approach to add collagen without using any harsh chemistry. And so these are simply mixed during the polymerization phase, and I found that it's working well for cell attachment. And also I'm seeing that just increasing the concentration of collagen is increasing lipid accumulation. So again, we'll dive more into this in future work, but it's interesting to speculate that perhaps more adhesive proteins within these scaffolds can affect the lipid accumulation as well. And so technically, these are completely edible. There is a little bit of rat tail in there, but if you want to wait, we're actually working to acquire some microbially derived collagen so that we'll have completely animal-free scaffolds. So in conclusion, towards efficiently engineering adipose tissue, I hope that I've shown you that scaffolds potentially can increase the efficiency of adipogenesis, and we can do things as simple as tuning the microcarrier size to reach this, and that the resultant engineered adipose tissue can be used as a building block to generate larger organized three-dimensional structures like a marbled cultured steak. I want to thank all of my funders, and especially New Harvest. Thank you so much for supporting me during my PhD. And everyone in the ROA lab, we are hiring a postdoc position. If anyone's interested, you can go ahead and scan that QR code. A huge thank you to all our collaborators. In fact, one of our collaborators, Rochelle Crosby, has joined us at the conference today. She is an expert in muscle cell biology, so if you'd like to meet up with her, I'd be happy to introduce you. And thank you so much to all of the team at ISCCM for having me. It's been a great honor to speak with you all. Thank you.
Thanks so much. I guess we have time for a couple questions. Who's in?
I have a question about the size of the beads, because did you use the same cell concentration for all the beads despite their different sizes?
Yeah, so I'm able to actually calculate the surface area using our protocol that we developed, and so I'm seeding according to available surface area for the beads.
Oh, okay. Mm-hmm. So it's not that if you would put more cells in the... Okay, got it. Because I was like, if you increase the cell concentration on the bigger beads to have the same surface area, if it would be different, but...
Potentially, yeah, I see what you're saying. But yeah, I tried to control for that with seeding density.
Okay.
Hi, Stephanie.
Hi.
It was a great talk. Thanks.
Thank you.
I just wanted to ask you about your earlier slide between day 7 and day 14 with your mouse differentiation. The adipocytes look very different. It looks like they were unilocular and then have moved towards multilocular. Do you have any idea of what's going on there, or what does it look like under the microscope as you're looking at that?
Yeah, I mean, I think we, noticed some variation kind of. So those were different samples since I had to fix them and stain them, so they're not the exact same cell. And so, yeah, we do see some of that differentiation. But I have noticed, I'm finding actually, I didn't show this data, but with the smaller scaffolds, I actually am seeing more, not necessarily unilocular, but larger lipid droplet formation. And so I'm not sure if that could be playing a role, but definitely something to look into.
Thanks so much, guys. I think we're actually just out of time. I'm so sorry, but Stephanie will be available to field your questions later, I hope.
Yes, I will.